And these are about the materials. Next to the principle. So coming to the principle, the Benedict's reagent of three chemicals like sodium citrate, sodium carbonate and copper sulfate, it contains Cu2 plus ions, that is copper ion in alkaline medium, that is in the pH above 7, and it is regarded as the basic, and this reducing sugar are oxidized to carboxylic acid. Yes. So this aldehyde group of aldose will oxidize to carboxylic acid, that is CHO plus O, that is oxidized to form carboxylic acid or enediol. This enediol is the reducing agent which gives the color to the result, and this enediol in alkali medium. So Cu2 plus ions are reduced to Cu ions, forming the insoluble cuprous oxide. So copper reduced into form cupric, and it just forms the insoluble cuprous oxide, and this cuprous oxide appears as the brick red color precipitate, which indicates the positive test of Benedict's test. And the reaction occurring while in the Benedict's test is reducing sugar, that is any type of glucose, fructose or mannose, plus copper, that is the blue color, it gets oxidized. Just oxygen level is added plus Cu2O, which gives the final product of red precipitation color. And this color intensity depends on the reducing sugar concentration. So whatever the sugar level is present, this color intensity will vary. So more the concentration of reducing sugar, more color intensity, and less sugar concentration will leads to less color intensity. And this is the overall principle which leads to the color formation from blue color Benedict's test reagent to red color, brick red color precipitate in that solution. And coming to the procedure, this is the procedure. First one, you want to add 1 ml of sample in test tube, and that sample may be of urine sample from diabetes mellitus patient or from normal patient, and or carbohydrate solution, that is glucose, fructose, any form of solution. In that 1 ml of sample, we want to add 2 ml of Benedict's reagent. So the sample and the Benedict's reagent will get mixed to each other, and further adding two samples of Benedict's reagent and sample, we must heat into the water bath or Bunsen burner. So we should apply the heat. While applying heat for 3 to 5 minutes, the color change will vary accordingly. So if reducing sugar is present, the color of the blue changes from green, and this green furtherly changes into yellow, and that yellow will lead to final brick red precipitation. So if the solution has sugar, reducing sugar in that, the color changes from blue to red. If the solution sample does not having sugar, it is in the form of blue color, no red color precipitation will form. So boil the solution according to the color changes of the solution. So any samples will vary from red, green, yellow or from blue. And this is the overall observation where you could see the blue color solution, no reducing sugar is present, and in green color only the trace amount of reducing sugar, and in orange moderate level of sugar, and in brick red precipitation you could observe the large amount of reducing sugar is present in the urine sample. And in this picture you could see in a Bunsen burner or in boiling water bath, the beaker consists of water, and that water has to be boiled. In that boiling water you want to add the test tube, where test tube comprises of test sample and Benedict's reagent, and it should be heated for 3 to 5 minutes to change the color from blue, green, yellow, orange and red. If the sample or solution does not having any reducing sugar property, it finally appears as blue. Even we are applying heat to that sample, the test tube color does not change from blue to red, it remains as blue. If it remains as blue, no reducing sugar is present. and furtherly the changes in color, the reducing sugar concentration will vary accordingly.